diretor científico da Fabetes, entre muitas coisas, para apresentar o Michael. Muito obrigado. Michael, thank you for coming.
Bom dia, pessoal. Então, eu sou amigo do Gustavo, da Maria.
You can speak closer to the
Yes. Can we take this?
No. It's
Okay,
this is the
Okay, all right. How do I go ahead?
O Michael é,
Here.
eu acho, o principal
Here.
pesquisador no mundo hoje em biologia de tumores cerebrais. Vocês depois podem olhar o histórico de publicações dele, vocês vão ver o quão impressionante é, realmente muito impressionante. E além das revistas que ele publica,
laser.
estudando, etc.
Low battery, but it's going to
Okay.
work for you.
Quase todo paper que ele faz muda a área. É um negócio assim, groundbreaking. É um negócio que realmente tem um impacto muito grande. Algumas das descobertas que ele fez, como características de grupos moleculares da doença, que eu e ele estudamos, que é Medulloblastoma, já tem consequência clínica. O tratamento das crianças é decidido com base na biologia do tumor, da forma como ele descreveu. E uma das coisas que faz o trabalho do Michael tão impactante é que ele é um biólogo molecular de excelência e um neurocirurgião pediátrico de excelência também. Então, ele está no lado clínico cirúrgico e traz contas de pesquisa, né, para a biologia molecular que são informadas pelos desafios clínicos que eles encontram. Então, isso faz realmente uma pesquisa de altíssimo impacto. Pesquisar depois do trabalho dele. Não existe nada de melhor nível em biologia de tumor cerebral no mundo feito hoje. Ele faz o melhor trabalho agora. E o Michael é um amigo da ciência, é um amigo pessoal, orgulhoso de dizer, é um amigo da ciência brasileira. Já esteve muitas vezes no Brasil, primeira vez no Rio, mas já teve muitas vezes no Sul, São Paulo, em Belo Horizonte. Sempre aceita os convites para vir, para palestrar, para fazer atividades com a gente. Já orientou uma tese de doutorado comigo lá na URGS. E é membro do meu INCT que eu coordeno. Nós temos um INCT de biologia do câncer infantil e oncologia pediátrica. E, com tudo isso, a razão principal de ele estar no Rio agora é que amanhã ele vai tomar posse como membro correspondente da Academia Brasileira de Ciências, né, como reconhecimento da academia, pela excelência dele e pela contribuição que ele dá ao Brasil, colaborando com a gente. Estamos sempre disponível para vir colaborar com a gente. Então, é um prazer ter trazido ele aqui hoje. Espero que é a primeira de muitas vezes no Rio. Está adorando o Rio, ele é sua esposa. Acho que é isso, né? Então, Michael, thanks for coming.
My pleasure. Good afternoon, everybody. Obviously, I'm going to speak in English, but you're scientists, so you probably all speak English anyways. I'm going to talk to you today about a brain tumor that I have been studying for about 25 years called medulloblastoma. In children, the most common type of solid cancer is brain cancer. And the reason for that is because the brain grows very quickly and then it's done growing, right? By the time you're 2 years old, your brain is pretty much done growing. Right now, the way we treat these brain tumors is by aggressive surgery, and then we radiate the entire brain and spinal cord, and then we give very high-dose chemotherapy. And still many of the kids die. And of the kids that live, as I'll talk about in a minute, they're very damaged. So we would like to find a better way. And I have been trying to find a cure for medulloblastoma and to understand it for 25 years. Now I'm starting to think maybe that's the wrong way to do it. And we don't try to cure colon cancer as much anymore. We try to prevent it. We don't try to cure cervical cancer. We try to prevent it. We don't try to cure breast cancer. We try to prevent it. Because we have learned as a species, as humans, that prevention or early treatment of cancer works much better than treating advanced aggressive cancer. There we go. So this tumor that I study, medulloblastoma, is a tumor of the neurons, right? The thinking cells in your brain, for those of you who aren't neuroscientists. And it was described 100 years ago this year, in 1925, by a neurosurgeon in Boston in the United States. Right? So this tumor has been around for 100 years, and I think it's time to get rid of it. 100 years is enough. Go away. All right? doing the right thing there? Yes. So humanity, our species, has known about cancer for a very long time. Even the ancient Egyptians knew about cancer. And through time, we have learned how to better classify cancers, right? First, it was cancer or no cancer. And that was like the first stage. And that goes all the way back to ancient Egypt. And then people started to notice that cancers in different parts of the body were different from each other. So a breast cancer is different from a lung cancer, different from a brain cancer, different from a big toe cancer, right? That was stage number 2. That was a very long time ago. And then we fast forward 100 years or so ago, started to look at cancers from the same part of the body by cutting very thin slices and looking at it under the microscope and seeing that this breast cancer looks different from this breast cancer, this lung cancer looks different from that lung cancer. So the era of histology. And that was era number 3 for classification of cancer. Probably, 100 years or so ago at the most. Then we moved into the era where we're looking at individual biomarkers, mostly by looking at proteins, right? And mostly immunohistochemistry. So these 2 cancers look the same under the microscope, but when I put this antibody on, this one turns brown, this one does not turn brown, so they're different from each other, and that was era number 4. And for a lot of the world, and many of the cancers, that is still where we are today. But we're now moving into the 5th era of classification of cancer by humans, where we're starting to use molecular biology. In fact, it's almost time. Oh, what did I do there? I have magic powers. Let's see if I can do that again. My magic ran out. So now we're into like the 5th era of classification, where we will use molecular biology. Maybe it's, yeah, okay. I'll put this down and I'll stand further away from it. And I would say that, the histology, the pictures of H&E under the microscope are very pretty, and we're not ready to get rid of them yet, but the end is in sight. The microscope is becoming old-fashioned, and we're going to move to molecular biology because we can understand things so much better. So, everybody knows here what a Bigfoot is, right? And you've heard of the Loch Ness Monster from Scotland and the Yeti from the mountains. I'm from the mountains in Canada. And this is a medulloblastoma. What do these things have in common? None of them exist. They are all things that were made up by humans, right? They exist in human heads, but these aren't really real. And there really is no such thing as a medulloblastoma. And the reason I say that is really, no, I did that wrong. No, I did it right. Really, there are several different diseases that look the same under the microscope, but when you use molecular biology, they're very different, right? The way we did this in the beginning was using transcriptomics, bulk transcriptomics. We just did the whole transcriptome of many tumors, and we said to the computer, which of these things are different from each other? And the computer said, boom, 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 and it said, okay, there's 4 different kinds. And when we went back and looked at the 4 groups identified by the computer, by bioinformaticists, we could see that the patients were different, the response to therapy was different, and many different things were different, right? And the 4 groups, the names are Wnt, sonic hedgehog, group 3, and group 4. The Wnt tumors, that I'm not going to talk about much more today, they have too much activity of the Wnt signaling pathway, right? That's why they're Wnt tumors. The sonic hedgehog tumors have too much activity of the sonic hedgehog signaling pathway. And group 3 and group 4 have boring names because until recently we did not understand them as well. Now we do. And the main reason they have boring names is when my lab discovered group 3 and group 4, we had good names for them, but the reviewers did not like the names because reviewers are boring.
What are the names?
I don't even remember at this point. Chinchilla and ocelot. I don't know. We're at the point too, though, by using more molecular biology, we can divide each of the 4 subgroups into even smaller groups. And what we have always suspected is that the groups, the different types of tumor, because there is no such thing as a real medulloblastoma, there's just 4 different diseases. Probably we see this and they have different biology because they started in different cells in the developing brain. Okay? A lot of cancer, the biology of it, especially the transcriptome, is not something that is gained by the cancer, it is something that it was born with, right? Most of you were born in Brazil. I was born in Canada. There could be a guy from India and a guy from Russia. You guys have a Brazilian accent because you're born in Brazil. I have a Canadian accent because I was born in Canada. A Russian has a Russian accent because they were born in Russia. It's very much the same for the transcriptome in cancer. Wherever you were born in the body, or which part of the brain and which kind of cell, that is carried forward to the mature cancer. Okay? So we talked about this a little bit at the beginning. Humanity has learned that it is much better to prevent cancer or to treat it very early rather than try and treat advanced disease, right? If you have colon cancer and you have the liver and the lymph nodes, even if you get very aggressive, you are probably going to die. If your wife nags you and you go get a colonoscopy and you remove the polyp, you will be all fine with no chemo, no radiation, no real surgery, right? So it's better to find the polyp than to find advanced cancer. Same thing for breast cancer. That's why women undergo mammography. Same thing for cervical cancer, right? Started with the pap smear, now molecular tests. And now there's even a vaccine for cervical cancer. Treating advanced cervical cancer in 2025 is still hard. If you show up with advanced cervical cancer, you're still probably going to die. But if you get the cervical cancer vaccine, you'll never get it. This is the real way forward, I think, for humanity, rather than trying to cure a very advanced disease. There's many more examples in oncology. And not only does this work better for the patients, right? It's cheaper for the patient, cheaper for the hospital, less painful for the patient, and less painful for the family. So this is something that is good for everybody, okay? And even if you could cure colon cancer or metastatic to the liver with massive therapy and big surgery and lots of radiation, it is very unpleasant. It's unpleasant for you and it's unpleasant for your family to watch you do it. And it's even more unpleasant if it's your 7-year-old boy, right? I want you to think for a minute about if you have children, about your 7-year-old child, or maybe your sibling, or your niece or your nephew. Imagine how sad you would be if you heard that they had brain cancer, right? And not only that, but then now they have to have all these terrible therapies and they still might die. So why don't we just avoid all of this by preventing it? If you get treated for medulloblastoma, the treatment is very severe, right? The patients are much more likely to have, as they get older, to have a stroke, to lose their hearing, to be on disability, so not have a job. They're very unlikely to be pregnant. They have a 20% incidence of developing cancer in the future. And after you get the radiation as a child to your brain and spinal cord, your IQ goes down 1 or 2 points every year. So the adults have an IQ of 60 or 70, right? Most of these patients, even if they survive, they live with their parents, right? They don't finish high school, have a boyfriend or a girlfriend, they don't have a job. So even when we win, it's not really winning. I don't like ties. I like to win. So we need to change the game. So this is the cerebellum. How many neurons do you have in your brain? Does anybody here know without cheating? How many neurons are in your brain? You're all using your brains right now. You're scientists. You're supposed to be the people who use your brains the most in the entire society.
86 billion.
86 billion. Did you cheat? Actually, I don't even have it on there. So yes, we have 86 billion neurons in our brain, which is a lot, but it's in some ways it's not a lot too if you think about it, because everything we do is done with those 86 billion neurons. As humans, most of us are very impressed with our cerebral cortex, right? The part on the top. Like if you see if someone wants to show a brain on TV, they show the cerebral cortex, right? What percentage of your neurons do you think are here in your cerebral cortex? Anybody want to guess? It must be most of them, right? Because the cerebral cortex does poetry and calculus and science, and all the impressive things that humans do are done by the cortex, right? But it turns out the cortex is only a small percentage of your neurons, 10 or 15%. Everybody put your hand on the back of your head. So for those of you who are neuroscientists, underneath your hand is your cerebellum, the little brain. It shouldn't be called the little brain, it should be called the big brain, because the cerebellum has 80% of your neurons, 69 out of 86 billion, right? So this one little area of your brain has most of your neurons. Medulloblastoma arises in the cerebellum. Do you guys know what Russian roulette is? Right? You spin the thing and you get lucky or you don't get lucky. Every time a cell divides, there's always some errors. You know, some single nucleotide errors, some copy number errors, every time, right? And every time a cell divides, if it has the wrong error, it can turn into cancer. So if you have 69 billion or 80% of the neurons in your cerebellum, every time it's making one of these, it's like playing Russian roulette, 69 billion times. And this is why medulloblastoma is the most common brain cancer, because the cerebellum played Russian roulette the most times. Right? Now you understand it. It's kind of simple, right? Exactly. All of these, and I'm going to talk about this more in a second, all of the neurons in your cerebellum are made by one very small part of your developing brain called the rhombic lip. And we'll talk about that in a second. So why is the human cerebellum so huge? We don't know the answer to this, right? And this becomes even more interesting if you look at all of these other animals, because underneath it shows the weight, and who cares about weight, but the number of neurons, right? So human brain here at 86 billion, 69 billion in the cerebellum. A mouse is only 71 million neurons for the entire brain. So your cerebellum has as many neurons as 1,000 mice. And in that mouse, it has to do everything. It's not just the cerebellum, it's the whole brain, right? It has to run around and eat and mate and take care of babies and hide, all that stuff, right? So your cerebellum has the same number of neurons as 1,000 mice. Who here thinks that, many of you probably use mice in your experiments, who thinks, because the cerebellum makes you have agility and dexterity, who here thinks they can catch one mouse? Has there ever had a mouse get away in the mouse facility? Really hard to catch one mouse. How come you have a cerebellum 1,000 times bigger and you can't catch one mouse? Could you catch 1,000 mice? Not ridiculous. So why is our cerebellum so big? We don't know. We can talk about this more if we have time. But even compared to our closer cousins, right? This is a monkey, a macaque monkey. So we are all monkeys, right? We are just slightly less hairy monkeys, but the more hairy monkeys, like a macaque, this macaque has only 500 million neurons, and he's a monkey. And even chimpanzees have about 35 billion neurons. So your cerebellum is the size of 2 entire macaque brains. So now you understand why we get medulloblastoma. It's because you're making this huge cerebellum, and you're playing Russian roulette, and everybody here, it was fine. It was always a blank bullet. But every once in a while, in one child's cells, there's the wrong mutation, and it turns into a cancer.
In terms of, can I ask you?
Yes, please.
In terms of percentage, the glial type cancer and neuron type, what's the for the glioblastoma, for example,
Yes.
compared to medulloblastoma? What's the percentage?
So glioblastoma is largely an adult tumor, and largely in the cerebrum. Glioblastoma in the cerebellum is super rare. I've been a neurosurgeon. I started my neurosurgery residency. I hate to admit this because it makes me sound old. I started my residency in 1994, 30 years ago. In that time, I think I had 1 glioblastoma of the cerebellum. It does happen in the cerebrum, but mostly in adults. Okay. For some reason, we could discuss this if we had to wait 2 hours. I'd love to talk about developmental biology with you, but glioblastoma is almost never in the cerebellum. Okay. Good question, Raphael?
Yeah, this is the reference used for the student.
Yeah. I'll send this to you later.
She used to be a professor at this university.
Oh, there you go. I didn't even know that.
Here's the place where this figure was made.
Okay, very good. It's a good figure, but at least I was smart enough to put the credit. Oh, and look, you know what? I should have put this for Brazil, but I have to admit I used it before Brazil. A human cerebellum is the same size as 43 capybara. Right? Which doesn't, it's kind of funny, but it's also kind of strange because as humans, we do these things that other animals don't do. But the other end, you know, they run around, they hide, they eat, they drink, they take mates, they have children, they raise their children, they teach their children, and they do it with a much smaller amount of neurons than we do.
The proportion is the same in the other animals?
I don't think that we've done that very well comparing the amount of cerebellum to cerebrum. What's interesting, and I don't have it on here, I think it's interesting at least, is, you know, elephants and porpoises have a brain that's almost as big as ours. And I think the elephant cerebellum is quite large. Probably I would guess that there are elephants that probably get medulloblastoma, but the elephant population is small. And if you're an elephant with a brain tumor, you probably just get eaten by the lions, right? The lions don't eat us humans as often anymore. The big difference between us and the porpoises and the elephants for thinking is that even though they have nearly as many neurons as us, our cortex is thicker, right? So we have more layers to our cerebral cortex. And I suspect that's at least part of the reason why we are the ones shooting the elephants and not the elephants shooting us. So this is a mouse, okay? And this is before neural tube closure, right? So the frontal lobes would be up here, okay? The spinal cord down here, and the neural tube is just closing. And in humans, remember, the neural tube closes around 1 month after mom and dad have a very good night and the egg meets the sperm. We are like a worm, right? First, we are a book, right? When we're a neuroplate, remember that from undergrad? And then the book curls up and we become a worm. And then the worm folds up into a human brain. And part of the reason that it folds up is different parts of the worm grow more than others. And the part that grows the most is the part that makes the cerebellum, right? Because it's making 80% of the neurons. And that's this part right here, and it's called the rhombic lip. See this blue here? And here it's outlined in green. And it's called the rhombic lip because it's around, see this shape here? It's like a diamond. In English, that's a rhombus. So it's on the edge of the rhombus. So this, who here remembers from undergrad the rhombomeres? Rhombomere 1, rhombomere 2, rhombomere 3 for the hindbrain, right? You all read about it, passed the test, forgot about it. It's fine, I did too. All of the cerebellum comes from rhombomere 1. So it's like the worm, and then you're going along the worm, and all of a sudden the worm gets really fat, right? Because it's making all the neurons there. And then the big fat part of the worm, it flops over like a beer belly on a man, and that's what the cerebellum is. The cerebellum is the beer belly of the brain. Okay, so this is the rhombic lip, okay? And that is where most of your neurons are born, and that is where medulloblastoma comes from, right? This is in a mouse. So when you hear me say rhombic lip, I'm talking about this. This is a nice 3D view. For the next sections we're going to see are going to be sagittal sections or axial sections through the neural tube. So back when I was working in Canada, we published this paper by my student Maria Vladio, who has her own lab now. And around this time, the Canadian Cancer Society was always saying to me, Michael, stop trying to, and I didn't listen to them, stop trying to cure cancer, start trying to prevent it. And I would say, you guys are stupid, because when you look like this, because this is, I think you look like this when medulloblastoma starts, all of you look like that. I want you to think of the person who you love, your mom, your dad, your wife, your husband, boyfriend, girlfriend. They all look like this at one point, right? But when you look like this, when medulloblastoma starts, you're not talking on cell phones and eating too many hot dogs. So how are we going to prevent medulloblastoma? In this paper, what Maria did is she sequenced with single-cell transcriptomics a bunch of mouse embryos, right, especially the rhombic lip and the hindbrain, and she compared that to single-cell sequencing of the tumors. And by doing that, we were able to find cells in early development from the rhombic lip that looked like the different tumors. So we were starting to be able to find the cell of origin for the different kinds of medulloblastoma. Sonic hedgehog, group 3, and group 4. We won't talk more about WIN today because it actually doesn't come from the rhombic lip. It comes from the lower rhombic lip, which makes the brainstem and not the cerebellum. So it turns out, and this is by a paper by my good friend Parthiv Haldapur, who's a neuroscientist in Seattle in the United States. No one had really looked before at the embryology, human fetuses of the rhombic lip and the cerebellum. And it turns out the human rhombic is very different from other animals. In other animals, as shown here in the mouse, the rhombic is this small little area and it looks quite bland. But what Partha showed is around week 12 after fertilization, the human rhombic lip splits into two. It splits into a VZ, which means ventricular zone, because it's close to the ventricle where the CSF and the choroid plexus are, and an SVZ, subventricular zone, which is further away from the ventricle. The ventricle part, the VZ part, contains the stem cells. As you can see, they're staining here for SOX2. Everybody sees the SOX2, right? Right? SOX2 is a stem cell marker. The SVZ, and stem cells usually divide very slowly, right? They're very slow and complacent. They give rise to progenitor cells in the SVZ, and this is Ki67, as you can see here. That is a fireball of mitosis in a human embryo 17 weeks after fertilization, because that is the area that's going to make the 69 billion neurons that are going to make up your brain, okay? This is a new discovery, the splitting of the rhombic lip. Probably only humans have a split rhombic lip. Partha has looked in macaques. He has not yet looked in bonobos or chimps, but I suspect this is only in humans. The other thing that Partha discovered, along with Kathy Millen, who he works with, is that in between the ventricular zone and the subventricular zone, only in humans, there are these vascular cells. Probably there's no blood flow through them, but they are vascular type cells, and they're only in humans. And in fact, I don't know if I have the slide here today. If you get rid of those vascular cells in humans, the cerebellum does not grow very well. And you get a malformation called the Danny-Walker syndrome, where your cerebellum is largely gone. So almost for sure, these funny vascular cells that are only in humans are making a stem cell niche. The stem cells are probably talking to the vascular cells are talking to the stem cells. This is probably part of why we're human, right? This is a human thing, and it's part of the human condition. So now this is just multiple times slide from part showing the black arrow is the rhombic lip. This is 30 days after section of the neural tube, and you can see the rhombic lip is just a little bit thicker. The red arrow is the GABAergic neurons, and GABAergic neurons don't make cancer, and they're boring, so I'm not going to talk about them anymore today. Sorry if anybody here likes GABA neurons. But if you watch the black rhombic lip, you can see here by 10 weeks, it's getting a little bigger. Now here at 12 to 13 weeks, it's split into two. This is the human-specific thing, right? And then later, it moves into the cerebellum, right? Because the cerebellum grows around it. And the cerebellum has hemispheres on the side, and in the midline, it's called the cerebellar vertex. We will memorize this for an exam once upon a time. The vermis has 10 lobes, 1, 2, 3, 4, 5, 7, 8, 9, 10. And all of the stem cells always move to lobe 10 of the vermis, also called the nodulus, which I didn't know until we started working on this. So the nodulus is where all of your stem cells go to retire. Like in North America, it would be the Florida. I don't know, maybe where do people go to retire in Brazil? In Porto Alegre, somewhere where it's cool or something? I don't know. To Rio, yeah. To Rio. Yeah. So Rio is the Florida of Brazil. But you see those moving. You see that dark pattern? Because those are the concentrated stem cells. This happened in all of us. But what happens in normal human development is all of those stem cells stop being stem cells, then there's only progenitor cells, and then they all differentiate into neurons. And around the time of birth, there should be no more stem cells, and there should be no progenitor cells, and just postmitotic neurons, right? Cancer is not postmitotic, is it? Cancer requires mitosis.
I'm going to skip this because I don't care about GABAergic neurons.
So we published this paper, if anybody wants to look at it, in 2022. And what we show in this paper is that the group 4 medulloblastomas, the most common kind, are really just a ball of the subventricular zone progenitor cells. They are very, very similar at a transcriptional state. And any way we measure them with epigenetics or DNA methylation, all of these things, a group 4 medulloblastoma is a ball of rhombic lip subventricular zone cells that are trapped at that fetal stage. They just can't finish, so they keep They cannot leave cell cycle, and they just keep making more and more until eventually it makes a tumor, right? These cells of the subventricular zone, they're mostly there in the second trimester of pregnancy, and we think it's the second trimester where group 4 medulloblastoma starts. I'm going to tell you in a minute that for group 3, the more aggressive kind that has stem cells in it as well, probably starts in the first trimester of pregnancy. So in fact, some of these cancers start before women even know they are pregnant, which is kind of mind-blowing, okay? And these are the cells, these progenitor cells, these glutamatergic progenitors of the SVZ, these are the cells that got big in humans to make our big cerebellum, right? So it's like Goldilocks and the Three Bears, right? Hot porridge too cold, porridge just right, porridge too hot. If you have porridge too cold, you get a chimpanzee brain, right? Small and you're not smart and you don't know how to write grant proposals. If you have porridge that's just right, you make the right number of glutamatergic neurons and you get a healthy human cerebellum like we all have. If, however, using the same mechanisms that expanded the cerebellum, it goes too far and you get too many of these progenitors and you never go away and they keep growing, what is that? It's cancer, it's a medulloblastoma. So it's really quite that simple. And I have had many students, postdocs, PhD students over the years, and we tried to make group 4 medulloblastoma in mice and it never worked. And it's just because mice have too small a cerebellum, right? So I will also say, we think now in my lab that human evolution is getting medulloblastoma in a small percentage of our babies. Okay? If you have a glutamatergic progenitor in the rhombic lip in a mouse, it divides once or maybe twice. So 2 to the 1 or 2 to the 2. So 2 or 4 neurons, right? That's why mice have a small brain. In your rhombic lip, when you were a baby, each of those progenitor cells divides at least 10 times, maybe 13 times. So that's 2 to the 10, which is 1,000 neurons, or maybe even to the. So now you understand how the cerebellum got big so quickly. They took this one kind of progenitor, stopped it from differentiating, and made more progenitors, and used the power of logarithmic growth, right? 2 to the 2, 2 to the 3, 2 to the 10, right? Every time you double, it starts getting very big very quickly, right? So this is now showing a section of the rhombic lip, right? This is the VZ where the stem cells were we talked about in yellow. In between is the funny vascular plexus. It's only in humans. And then on the other side, if you move from here to here, you become a progenitor and you divide like crazy, right? After that, you make a choice to either go on the surface of the cerebellum, where you divide again, and something called the external granule cell layer that makes the IGL cells of the cerebellum. This is the only area in your brain where you make a lot of neurons after birth. And in fact, you're still making neurons up to about 18 months in a human baby, right? Most of the other areas in the cortex and the other unimportant parts of the brain, that's a joke, most of your neurons are done, right? And some of your neurons decide to become unipolar brush cells, another kind of glutamatergic neuron, right? So this is now data from Kim Aldiger, who works with Parthiv and Kathy in Seattle, human fetal cerebellum studied by single-cell transcriptomics. You start out as this yellow stem cell, and then you become a progenitor cell, and then you decide to go out here to become an EGL cell, or up here to become a unipolar brush cell. So it turns out, now that we understand all this at a single-cell level, and we understand that the human is different from the other animals, from the more hairy monkeys and the mice, that a group 3 medulloblastoma is from here because it has these cells. A group 4 medulloblastoma is from here because it has those cells. And a sonic hedgehog medulloblastoma is a tumor of one of these, right? So it's really quite that simple. We couldn't understand it all before, and they all look the same under the microscope, right? Remember the slide about the Yeti and the Bigfoot? They look the same under the microscope, small blue cells. But now we can understand by molecular biology the 3 different types of cancer are from 3 different cells of origin. Yes?
So, if I understood correct, when the cells commit to be glutamatergic neurons that they get the cancer phenotype, or the progenitor cell like nestin, it's already undue for the process of cancer.
So the rhombic lip is rhombomer 1, right? We talked about that. In rhombomer 1, it is 100% glutamatergic neurons. It doesn't make any astrocytes. It doesn't make any oligodendroglia. It doesn't make any GABAergic neurons. Every single cell in there is committed, right? And as far as nestin goes, I'm not going to talk about it today, but we have it in a paper we published in Cell last August. Nestin actually turns on probably around here in the progenitors. Nestin is too late. Because these cells, they're very early, like first trimester, right? And they use different markers, and I think we're going to talk about that a little bit later. Rhombomer 1 is very weird, right? Because almost every other cell in neural tube makes neurons, and then it makes oligodendrocytes, and then it makes astrocytes. Not rhombomer 1. It wants steak, steak, and steak, right? It's only glutamatergic neurons. So now you understand how simple this is, right? Stem cells make group 3, progenitors make group 4, and granule cell precursors make sexual tumors. 1, 2, 3. No problem. And in fact, remember I told you that all of those stem cells move to lobe 10 of the vermis, the nodule, right? And when we now look at the tumors, so you can see this is a brain, obviously. This tumor here, it's coming out of the nodulus. And when we look at all the other tumors, it's now very obvious in retrospect that the tumors all grow out of the nodulus. So now I'm going to say something. I'm going to point something out. I want you to think. If the tumor started in the first or second trimester of pregnancy and it was in the nodulus, what percentage of the patients must have cancer already on the day they're born? All of them. It was already there when they were born, and it was probably small. So we talked about this earlier in the talk. Is it smart to treat the giant metastatic tumor, or is it smart to treat the early small tumor, right? So theoretically, these things should be there before, because everybody has stem cells in their nodulus, but for all of us, they went away. But a kid with medulloblastoma, it doesn't. So at the time of birth, these kids must have them already there. So this is just to see it a little bit better, okay? OTX2 is a transcription factor that is highly expressed in medulloblastoma. I'm using it here as a marker. It is also an oncogene, but that's not germane today. You can see here's the rhombic lip staining very, very strongly. It's split early on at 14 weeks after fertilization. Then it moves here into the nodulus. And yet already by 19 weeks, you can see it's largely gone, right? And by birth in most humans, it's gone. The stem cells and the progenitals are gone, right? So it turns out way back in the 1880s, in the German language pathology literature, pathologists are already seeing funny stuff in the nodulus in some babies and some adults. And then later in the 1940s, pathologist from the Mayo Clinic in Rochester, United States, saw some funny stuff in the nodulus. And he said, I think this might be the start of medulloblastoma. The people ignored him. And then again, 25 years later, a pathologist named Lucy Rourke from the Children's Hospital of Philadelphia said this again. She said, I think that this is probably the cell of origin for medulloblastoma. Look at this. And again, people ignored her, partially because she was a woman in the 1960s, partially just because everyone thought the medulloblastoma came from the EGL. But look at this case. This is a case of an autopsy. Here is the cerebellum, and here's like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here's the nodulus. The ventricle, here's the brainstem. Look at that in the nodulus there. It's a collection of small blue cells. What did medulloblastoma look like on those sections I showed you? A collection of small blue cells. So this is a pre-medulloblastoma. So just like a colon polyp is to colon cancer, this is a cerebellar polyp in the nodulus is to mature medulla, right? It just stuff right now that it's not causing any problem. Later on, it will get big and cause a problem. Okay, so we call these things now pearls because it's the rhombic lip, right? And it's persistent because the rhombic lip should go away. If your rhombic lip lasts after birth, it's a pearl. So it's a pearl. And we named them after Lucy Rourke, who described them in the 1960s, so we call them Rourke's pearls. This is what your normal rhombic lip looks like before birth. You can see there's little islands of stem cells persisting and just about to fade out and go away, right? And this is another autopsy showing now pearls in a kid who is an infant, okay? And what's very interesting when we look very closely at the pearls, or in fact at the resolving rhombic lip, is that the stem cells and progenitor cells are all clustered around those funny vascular cells. And remember, the funny vascular cells are human specific, and they're part to help expand our human cerebellum, right? So this is very interesting now, isn't it, from a developmental biology and an oncology standpoint. Now, some of these pearls grow up to be medulloblastoma, but we know now that it's not 100% of them. And we know that because a pathologist from Stanford shared this slide with me of an autopsy. This guy was 102 years old. He was living in the Veterans Administration Hospital in the United States, and I think he had dementia. And he wandered outside and he got hit by a bus, right? And then they did an autopsy on him, and he has a pearl. And clearly it never bothered him, right? If you live to 102 and get hit by a bus, you did not die of medulloblastoma. So this means not everybody with a pearl gets medulloblastoma. It's not 100%. So what we need to know now is what percentage of people who are like 6 months or 1 year old who have a pearl will go on to be a medulloblastoma. You know, if it's 1 in 1,000, then probably there's nothing we can do about it. I have some friends who are pathologists, and they are now routinely taking the nodulus on all of their autopsies and looking for pearls. And so far, I think they found 0. So while not 100% of pearls turn into cancer, it seems like most of the pearls turn into cancer. Just like not all colon polyps turn into colon cancer, right? But having said that, we know not all colon polyps turn into colon cancer, so we should just ignore them, right? No, that sounds stupid. And I think it's the same thing for here. So now we're in a situation where maybe we can start doing early treatment, right? Or some people call it secondary prevention of medulloblastoma. Can we find these kids that have pearls and treat them early before their tumor gets large, before their tumor gets metastatic, so that when we're taking it out, we do less damage? And maybe so that we don't have to give radiation and chemotherapy, just like with a colon polyp. Can we switch medulloblastoma from a pediatric oncology problem to a public health problem? And that is what I'm trying to do in Texas. In Texas, we have 300 to 400,000 babies a year. And we're working now to make a blood test to scan all of them, right? Right now in Texas, I don't know about Brazil, every baby that's born has about 50 tests, so mostly to look for inborn errors of metabolism. Many of you will have heard in undergrad about phenylketonuria, or PKU, right? If you find that early on, you change the diet, the kids have a relatively normal life. You don't, their IQ goes in the bucket, right? So I want to find all of these kids. we're going to invent a test in Texas. We're going to scan all of Texas, and we have it working. We'll share it with the world. Okay, we'll talk about how we're going to do that in the next few slides. So this is a paper now. I talked to you only about group 4 before. Now I'm going to talk to you about group 3 medulloblastoma. This is a summary of it. So the group 3 tumors come from the stem cells, the yellow stem cells in the ventricular zone. But when we look inside the group 3 tumors, we see lots and lots of those funny blood cells, those blood vessel cells. We don't see them in group 4. Okay? So the tumor is mimicking normal development so that the stem cells can persist over time. So if you have a normal rhombic, by birth it all goes, all the stem cells go away and the funny blood vessels go away and you get a nice normal human cerebellum. Mama bear cerebellum, right? If you lose the funny vascular plexus, your cerebellum doesn't grow properly. It's hypoplastic and you get something called Dandy-Walker syndrome, which is actually quite common and something I see quite frequently in my clinic. If you just have progenitor cells that stay in the nodulus, you get a group 4 tumor. If you have stem cells, VZ cells, that are supported by the funny blood vessel cells, the interposed vascular plexus, then you get a group 3 tumor. That's the answer. And now that you see it, it's really kind of simple, isn't it? So again, what I just told you was a group 3 tumor is not just these VZ cells, it's a mixture of the VZ cells and the funny blood vessels. So there was an argument many years ago about the cell of origin. This is like the 1940s, the 1960s, the cell of origin for medulloblastoma. Most people thought it came from the EGL cells, that we talked about on the surface of the cerebellum that continue to divide up to about 8 months after human birth, right? And I can see why they thought that, because most of our autopsy specimens are not from very early fetuses. It's even rare to have a baby. The baby is more common, unfortunately, right? So most of the people thought that these EGL cells were the cause of medulloblastoma. There were papers, as I said again, first from the Germans, then from the Mayo Clinic, and then from Lucy Rourke at CHOP, saying that these were. And it turns out that both groups were right. Sonic hedgehog tumors come from the EGL. Group 3 and group 4 tumors come from here. And I've kind of alluded to this, but when we published, when my student, my PhD student, Liam Hendricks, made all these discoveries and we were writing up the paper, I said, Liam, I have very bad news for you. You were scooped before your parents were born. Maybe you were scooped before your grandparents were born, right? Because people had seen these a long time ago, but they just didn't understand it at the time because they didn't have access to single cell sequencing. They didn't have access to everything we know now about the differences between human and non-human cerebellar development. So this is the paper we published, and I just want to read this one statement to you. We published this in a relatively good journal, and the fact that I got away with saying this means that it's probably true, right? As prevention is preferable to cure in the future, we predict the development of postnatal screening tools to detect pearls across large populations. I think that's the future for my field. And right now I'd like to tell you there's probably 3 different kinds of pearls because we're starting to find some of these things incidentally. When we're looking at the MRIs of kids, a kid comes in with a medulloblastoma, and maybe that kid fell at the playground and had an MRI or a CT a year or 2 before. And when we look back, we can see it. So that's an incidental pearl. And I think going forward, there will probably be some kids who fall at the playground or have a minor car accident, and then we will see these prospectively, and we'll have to decide what to do. But my ultimate goal is to make this a public health problem, as we'll talk about a little bit more, and find these kids around 6 months of age and treat them all, treat them just like you treat a colon polyp. Okay, so this is a case published in a paper by Smith et al. In Nature in 2022, not my paper. This child came in with a medulloblastoma, group 3. Everybody can see it there. If you look back in time, 9 months, this is the nodulus, and you can see that it's asymmetric and it's too large and the signal is wrong, right? So this pearl was already there and visible months ago. I would contend that maybe if you had taken this out 9 months before, maybe it wouldn't be metastatic, maybe you wouldn't have to do all the other therapy, right? So that's just one example, Michael. Anybody can find one example. About this one. This kid, again, medulloblastoma, right? And this one, you even see the metastases along the spinal cord. The kid a year before had an MRI that was read as normal. Do we think that's normal? Look at the nodulus. You can see the pearl. You know, of course, none of this stuff was published at the time, so I understand why the radiologist missed it, but it's very clear there, isn't it? Okay, 2 examples. 3 examples. Another medulloblastoma, 4th ventricle, right? Look, can you see the nodulus is bigger on one side? It's asymmetrical. Another pearl. Here you go. Example number 4, medulloblastoma in the 4th ventricle. Look at that nodulus, pouching out and overgrown into the 4th ventricle and changed in signal from the rest of the cerebellum. So these things are real. And in fact, though, I really want to find these when they're even smaller than this, right? And I don't think that the way, I said there's 300,000 or 400,000 babies a year in Texas. Now in Texas, we have a lot of resources. It's one of the many reasons I moved there. But even Texas can't afford to do an MRI on all 400,000 babies, right? And plus there'd be problems because they'd have to be sedated and all that. So I think this is not the way we're going to do this at a public health level. I do think in the future that kids are going to have minor trauma and stuff, and we'll find these and we'll have to decide what to do with them. But to find these across populations, it's probably going to have to be a blood test. Not a CSF test, because I also don't think we're going to do 400,000 lumbar punctures. It's just not going to happen. But we need a very good blood test. You all understand the difference between sensitivity and specificity. I want a sensitive blood test. So I'm okay with some false positives, but I don't want any false negatives. So I want to make a blood test so we find everybody and then bring them in and probably do additional tests. So one of the things that will be important if we do a blood test is what is the blood test going to be for? Now, as we all know, most cancers leak stuff into the blood. And in fact, for most patients with cancer, they have tumor cells going around in the blood, right? So if you have a pearl, I think in a normal person, it goes like this. The pearl's there and then it goes away, right? Around the time of birth, maybe a couple of months. But if you have a pearl, does it just sit there, or are there cells being born and dying, right? In most cancers, there are many cells that die, right? It's just that the rate of new cells is slightly higher than the rate of cell death. And over time, it accumulates. And I'm hoping this is what also happens in pearls, because if there's cells being born and dying, there'll be stuff leaking out into the circulation, right? And when do the cells that make a pearl, they come from the first trimester and the second trimester. And luckily, they use genes that encode for proteins not used after birth. Right? So if those proteins that are fetal proteins are found in the blood, then we can have a high suspicion that person has a pearl. And this has been in other cancers, right? So-called oncofetal-proteins. In fact, there are some blood tests that already do this, looking for proteins. Then the question becomes, well, how does a pearl turn into a big cancer? Right? Is it just slowly over time? It doesn't look like it on those things I showed you, does it? It looks like they were small, and then all of a sudden, for a period of time, they got big. How big? I think it's probably from additional mutations, right? Especially if they are cycling, as I show right here, right? They're cycling, they're an adenoma. Then all of a sudden, in one of those mitoses, one cell gets an extra mutation, and it starts to accumulate. Either because it's dividing more quickly, or there's less cell death. In that, I talked about bad hot dogs and cell phones, and I said I thought it was BS. Now I'm not so sure, right? Because in the past, when people have done molecular epidemiology studies of medulloblastoma, the control group was usually all the normal babies. I think now we need to do that molecular epidemiology comparing exposures and viruses only in the kids that have pearls, right? Because if you don't have a pearl and you get CMV, it might be different than if you have CMV and you have a pearl, right? So maybe bad hot dogs and viruses really do matter. I will point out, and I know this is a very speculative idea, the kids who get group 3 and group 4 blastoma usually present when they're about 5, 6, 7, 8 years old. And what happens to children when they're 6 years old? They go to school. What happens when they go to school? They're sick all the time because they get viruses from their friends. And this is really a coincidence, but a very interesting coincidence. So,
kids go much earlier in the nursery, right? I mean, they
true. Yes. In some places, yes.
Here.
Yes, absolutely. Some may be completely wrong about the virus idea. I just think it's interesting, and I love big ideas. So, and you certainly are exposed to a lot more children when you go to school, and even we know that as pediatricians, if you talk to the pediatricians, when the kids go to school, they start catching all of those funny viruses that kids get, like Coxsackie virus and hand, foot, and mouth disease, all that stuff, because they're all interacting with each other and slobbering over each other. Kids are kind of gross, really, right? Especially if they're not your own. So how are we going to get to prevention? We need to identify a biomarker, probably in the blood, right, to find the pearls, and then treat them, just like we would a colon polyp. So how are we going to get there? Again, I talked about this before. I think it will be a blood test. If you say there's 300, people always are going to say to me, I have 2 slides on this because it's important. Michael, that's so expensive. That's a nice idea, but oh my goodness, just 300,000 babies? From what people tell me, these perinatal blood tests usually cost about 10 cents a kit, right? Up to $1 a kit, right? Let's say it's even $1, right? So 300,000 babies at $1 each is $300,000. There are surgeons in Texas who charge more than that to take the tumor out, right? So if we do this and we find even 1 kid, we break even. We find 2 kids, we save the system money. Forget about all the misery and stuff we save, but I think we will also save money. In the end, after we invent this in Texas, we want to share it with the world. There is about 100 million new humans on this planet every year. right? 140 million. So if we could prevent medulloblastoma in the world for $140 million a year, that would be a great deal. That is way less than we spend on colonoscopy in the United States alone on colonoscopy, right? So $140 million a year, that's nothing, right? So we can do this just like we did for polio and all of these other diseases. Skip that. So there's two ways we want to find and make the biomarker. One is to do single cell sequencing and looking at things that are in the tumor that should not be present in a placental baby, like expression of proteins from fetal development. That's what I'm doing right now, and we're doing a lot of sequencing to figure that out. The other way to do it, though, is to take blood samples from 1,000 children with medulloblastoma and 1,000 controls, and then give them to people with fancy proteomics machines and stuff like that, and just say, how are these different? And in fact, people are doing this right now for a lot of other cancers to develop biomarkers. So we are gathering up blood samples just starting now around the world. And that sounds like, oh, Michael, how are you going to get 1,000 blood samples? There's a company in the United States now that invented so that kids can take their own blood at home. Kids can take blood, not their mom. It's this machine, and it has a whole bunch of very small needles, and they're very shallow. And when they put it in, pain because they go so shallowly, and it sucks up a capillary, and you get about 0.5 ml to 1 ml of blood. And I've seen videos of these kids and like, ah, ha, 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 boom. And these kids are doing it every week. And we're going to do this. We're starting it first in the United States. And the second country to start this, you know what it is? It's you here in Brazil. Done by Allegra, like Raphael, and some other people, because you guys have 200 million people here, right? So we're all the big countries together, and we'll work together as a species. We slightly less hairy monkeys, and we'll get these 1,000 blood samples, and we'll compare them to 1,000 controls, and we'll say, aha, that peak is different, and we'll make a test. Yes.
But the children with medulloblastoma diagnosed might have much more differences in the proteome than the
They might. You're absolutely right. But when we look at the medulloblastoma cells, right, they are very similar to the rhombic lip VZ and SVZ. An adult cancer, the way adult cancer works is you have a nice cell. Right? It has 2 kids, a mortgage, a Volvo, goes to work every day, and then you put 100 mutations or 1,000 mutations, it turns evil, right? Medulloblastoma usually has 1, 2, or 3 mutations. So it's very similar. So we don't think that the cells in the pearl are very different from the cells in the medulloblastoma. I admit there might be differences. It's true.
But like other way would be just to get a cord that you can scan and make the proteomics from that cord that has the pearl.
Yes, but how are we going to find the pearls in the first place?
Yeah, you have to.
Right? We're going to find 1,000. So I think the easiest way to do it is to start with the medulloblastomas, recognizing that maybe a peak that we found might not be present in the pearl. I fully take your point. I suspect it will, because I think the big difference here is a fetal cell type compared to all the non-fetal cell types. And I'm hoping that will make it easy. I'm not going to do this, but because can you imagine, right? You guys know what an IRB is? You guys, when you ask for permission from the institution to say something cool, and you have to do this much paperwork to do something silly, right? Imagine I had to do that in 1,000 hospitals. I would be dead, or I would wish that I was dead before it got done. So instead, this is going to be done by parents. Parents of children with medulloblastoma are going to do all the work and gather up these 1,000 medulloblastoma blood samples, and then they are going to give it to various scientists, not me, because I'm not a biomarker scientist. I'm a genomicist and a developmental biologist. And then we will find the peaks and we'll make the biomarker. These parents have had some of the worst thing that can ever happen to you, which is watching their child die. And not watching them die, but watching them die slowly, right? So they have a lot of sadness and a lot of energy, and they're the people, these parents, mothers, they're the people that are going to make this problem go away. We're going to change the paradigm. We're going to change medulloblastoma from a pediatric oncology diagnosis to a public health diagnosis. And we already talked about price. I just want to point out again, there's 140 million babies a year in the world. If that was $1 each, that'd be 140 million US dollars. In the United States, we spend $43 billion on colon cancer screening, right? So that is a very small fraction. Right? We can afford $140 million. That's a record at this point. So what are the problems from the idea? Not all the pearls turn into medulloblastoma. So we need to find out what percentage of them do. If every second person in here had a pearl, we couldn't do something about them all. I don't think it's true. So we're doing a natural history study. There could be things in the nodulus that mimic pearls. I don't think that's true. You know, I've been neurosurgery for 25 years. I don't ever see anything in the nodulus. What if the pearls are already metastatic? That would suck. And it may be true, but that just means that the treatment for the pearls can't be local surgical. It'll have to be, you know, some kind of therapy injected into CSF, or hopefully an therapy, right? Will it be too hard to develop the blood test to screen for pearls? We have many blood tests for cancer now, right? Who here has heard of PSA, the prostate-specific antigen test, right? We don't use that anymore. Do you know why we don't use it? It's too good. You know, it finds prostate cancers like 2 or 3 cells. All men get prostate cancer if they live long enough. We don't need to know about it if it's only a 3-cell cancer. Because the technology is so good, we can use the same technologies that have been used in the adult cancer once we have our biomarker, a protein, or a metabolite, or a mutation, et cetera, et cetera. What should we do once we find them? Should we treat them or observe them? I don't know the answer to this. It probably should be tested in a clinical trial. I have to say, if it was my kid, I would just take it out. Because if you take out the nodulus, I can take out a baby's nodulus, and they'll go home in a few days, and they really won't be much different as an adult than if their nodulus had been normal in the first place. Could we inject something, some drugs or some poison in the nodulus? We could. Could we do immunotherapy? Yes, we could. My lab has already invented a CAR T cell that works against the RLVZ cells. And it works very well in mice. And my colleagues as children are now prepping that CAR T cell to take to clinical trials for kids with full-fledged medulloblastoma. But in the future, it could be used, and other immunotherapies could be used to treat kids with pearls. But ultimately, my dream is to make a vaccine.
The messenger RNA that Pfizer used for COVID, this is more or less the technology that we're using.
I think that might be a way to do it. Messenger RNA vaccines are not very popular right now in some parts of the world, but yes, I think that might be the way to go eventually. And the difference, many have probably heard about that pancreas paper that was published a while ago where they made individualized vaccines based on mutations. I think here we can make a vaccine based on proteins that are used in utero in the rhombic lip, in fetal life, but not used postnatal. And that way we can use them for everybody. So my dream is, you know, we test all 140 million babies in the world, and if you have a pearl, or it looks like you have a pearl by test, we give you the vaccine, and we say, come back in 3 months and we'll repeat the test. Right? And we'll just make the whole problem go away. We'll put medulloblastoma into the textbooks and the history books. Right? I think that our species will still find medulloblastoma fascinating 200 years from now. It will be in all of the neurobiology books because it has taught us a great deal about the normal development of the human hindbrain. But it'll be it'll be something interesting. You know, scurvy taught us vitamin C. Rickets taught us about vitamin D. ALS, or amyotrophic lateral sclerosis. You guys know what that is?
Yeah.
ALS taught us about anterior horn cells. I just wasn't sure what it was in Portuguese, right? And medulloblastoma taught us about hindbrain development. After surgery for medulloblastoma, kids have this problem. Even if you do the surgery perfectly, they stop talking. And then later they talk again, but their IQ has gone down. We think it's from scraping around the tumor. And this happens in about 30% of patients. I'm hoping, though, that if we treat these as pearls, that will go away, right? That's the hope. And I'm almost done here. I'm going to skip that. I'm going to skip that because I'm a bit late, although, to be fair, we started late. Conclusions. Group 3 and Group 4 medulloblastoma are prenatally. Every kid who's going to get it is born with the cancer. We started in many cases before they were pregnant. It's in the nodulus of the cerebellum. If we remove or treat it, I think we can prevent or do like at least early treatment if you prefer medulloblastoma. We could avoid the cost of therapy. I think we can increase the cure rate of the healthcare system. And be able to avoid all that aggressive therapy. that might avoid this posterior fossa mutism. We're seeing these in real time. Some kids are going to see these now prospectively, right? A kid's going to fall off a bike somewhere and get an MRI, and the surgeon's going to see it, and they're going to have to decide what to do. Even before we make our biomarker and start screening kids around the world. And I think this is one of the few chances in the world where surgeons can actually make their lives better of children with cancer. And I think it's a way to change the world. This is my lab in Toronto before COVID, and this is some of my new lab in Houston, Texas, and I thank you for your attention. Questions? That's only the external granular layer.
So you wouldn't have.
And that never gives rise to group 3 and group 4.
So you could do like when you want to speaking with this proposed blood test for the
I think you could at 6 months, all of the VZ stem cells and the SVZ progenitor cells should be gone. We could also try to prevent the sonic hedgehog tumors that come from the EGL, but with that blood test or whatever would have to be done probably after 2.5 years. And the problem with that is that a lot of those patients present as babies, so it won't be as easy. I'm starting with the easy one. Always start with the easiest case. Lowest hanging fruit first.
Because very young babies, they are brought to the hospital all the time, they need to get vaccines and
Yes.
so they would be easier to get access to them.
To get the blood,
Yeah.
it's even easier if they do it in their own home, right? Brazil, I imagine, is a lot like Canada and the United States. A lot of patients live, an hour or more from the hospital. This machine is so easy. The kids do it themselves. They go, boom, right? There's a number of studies going on right now where they're collecting blood from kids with a number of different conditions, and they're taking the blood every week. Mom doesn't mind, kids don't mind.
Hi, thank you very much for excellent talk. It was very brilliant to see all of those stuff. And I am very curious about the choroid plexus. And
I skipped that for
in the fourth ventricle, you like a very good reach population of choroid plexus, and you have a lot of vascular cells too. And you were talking about this vascular niche, right?
Yes.
So the rhombic lip is very close to the rhomboid fossa.
Yes.
Right?
Yeah, actually, the rhombic lip is right beside the choroid plexus. And you probably know this, but for everyone else, in the supratentorial brain, everyone knows that the choroid plexus secretes a number of proteins that feed the radioglial cells that are the stem cells of the supratentorial brain. We suspect the same thing is happening in the fourth ventricle, that the choroid plexus is feeding the RLVZ cells. And in fact, if we take choroid plexus and we grow it with medulloblastoma, it grows better. So we think the same thing is happening. And we think that's probably even part of the reason why medulloblastoma metastasizes the way it does. The snack on the things made by the choroid plexus. Yes. Yes.
And one of the main difficulties is that sometimes we test 100 samples and we find a positive 5, which doesn't mean that the tumor doesn't have the positive.
Yes.
It's quite hard. So how are we going to overcome the problem with analyzing blood and what sort of biomarkers?
Yeah, so you're talking about the problem of false positives.
Yes.
So I'm not a biomarker person. But the way I see it is we initially make a very sensitive test, and we eliminate 99% of the population. And then we bring those people into a second test. That might be by another blood test. It might be by a CSF test, right? And then with CSF, we could probably sequence it and look at multiple genes, or we could even do imaging like MRI. So I don't know the answer, but the stakes are high, right? So I think someone is going to figure it out, and I think some rich people are going to give me enough money so that people who do this work can figure it out for me.
Any more questions? I think I have one.
Yes.
So you're trying to find out a biomarker,
Yes.
particularly a specific one. But are you trying to find a marker in cells from the neighborhood? Because maybe donor cells can secrete stuff and modify the cells in the mixture, and those cells start to secrete something else, something important that could be used also as a biomarker. And maybe you need to use a panel of biomarkers. So are
I'm sure it will be a panel.
Yeah.
Ultimately, I'm sure it will be a panel. We've thought about this idea. I think it's a great idea. Remember we talked about the funny human-specific blood vascular cells and stem cells and gender cells?
Mhm.
They're kind of messed up, right? And they're not present postnatally. So, example, something secreted by those cells could also be a biomarker. I agree with you 100%. Yes.
Any more questions?
I have a question about if concussions can increase the chance of this type of cancer occurring.
I don't think so. Concussion is usually mainly involving the cerebrum, on top, because there are twists, right? I don't think the cerebellum twists the same way as the cerebrum does. And if you think about it, medulloblastoma, many of them are babies or toddlers or young children. Concussion is much less common in those because you have a mother who watches you. Concussion is usually in stupid young male teachers and stupid young teenagers, right? So it's probably not as much. There is some very good work done by my former colleague in Toronto, Peter Dirks, suggesting that brain trauma might influence and contribute to cerebral or cortical glioblastoma. Probably less important in these babies, though. You know, in English we say treat it like a baby. Be very careful with it. So, and I think that's what happens. So the incidence of a head injury, particularly a concussion kind of injury in babies, is much smaller. Thank you very much, everyone.
Thank you.
Quero comentar só que tem um NCT, de câncer infantil, que eu coordeno, ajudar colegas pesquisadores, e tem um centro do NCT aqui no FJ, que é coordenado pelo Sérgio Anjo, que era o gestor, agora eu faço parte do NCT também, todo o time de vocês, Evandro Sobral, tem dado uma contribuição fantástica, né? O NCT também trabalha com todo o lado tecnológico, uma série de coisas do então nós estamos aí também.
Can I yank this? Yank? Thank you.
Very much.
I'm always afraid I'm going to forget it.
Great thought. Thank you very much.
That's very good. Yes, so that's very interesting. Yeah. So the only my place about the newborn screening is something that